Welcome to another seminar of our seminar series at the Irving Institute for Cancer Dynamics. Simon today is away traveling, so I have a pleasure of introducing our speaker. We'll have a talk by Jingyi Jessica Lee, who is an associate professor at the Department of Statistics and the Department of Biostatistics, Computational Medicine, and Human Genetics at UCLA. And she's also a Harvard Radcliffe Fellow in the years 2022 and 23. She got her PhD from UC Berkeley and her first degree from Tsinghua University in China. And Jessica today will present some of her research, recent work, which focuses on enhancing the statistical rigor in genomic data analysis and and she's aiming to develop realistic simulators for benchmarking and quality control of single cell sequencing and spatial omics data analysis. Thank you, Jessica.
Thank you, Carol. And thank you very much for the kind introduction. It's my honor here to present our work here today. And I'm from UCLA, but this year I'm staying in the Harvard Radcliffe Institute to write a book, actually, which is very related to my talk here today. So for this talk, I will focus on our recent efforts in the simulator development for multiple purposes, including benchmarking computational methods to make statistical inference from data and to design in silico controlled experiments to ensure the quality of our data analysis. So I will briefly introduce what the data structure look like. I try to summarize single cell and spatial omics data in one framework. So here the process data will be a matrix, which is the main information. It's a cell by feature matrix. Some people may write it as a feature by cell matrix as a transpose, but you can just consider this to be a matrix with non-negative values usually, or also mostly counts. And also we may have some covariates, which you can consider them as some additional variables of cells. And these are optional information we could use. So here the data have some heterogeneity structures about cells, which are rows of this matrix, and they could be defined based on cell covariates, or they could be latent structures we need to infer. So for example, the cells may belong to discrete cell types or clusters, depending on whether they're known or inferred. Or the cells may follow continuous trajectories, and this is usually unobserved, treated as a latent structure. Or we may measure the spatial locations of cells. And this is known for spatial omics data, but unknown for non-spatial omics data. If you just have single cell data, usually we mean you don't have the spatial information. And also the data could have experimental designs. So for example, we may have cells measured in more than one batch. So here the batch effects are unwanted effects. We want to remove those. Or we may have cells measured from two conditions, like cells from healthy donors and cells from diseased patients. So here the condition differences is the biological signal we want to infer about. And finally, the data may have different types of features. The most common one is gene expression, and this can be measured by single cell RNA-seq, single cells short as SC, or spatial transcriptomics. So these technologies give us gene expression at the RNA level. We could also have additional features like chromatin accessibility, which means that we could know which genomic regions are open. And this can give us the transcriptional potential of those regions, or how genes expression is regulated. And we may also have protein expression levels. So all these technologies are giving us different dimensions or different perspectives about those cells. So the motivation of our work is, first, there is a great need for computational benchmarking. Just from this website that collects computational tools for analyzing single-cell RNA-seq data, we have more than 1,000 available. So then from a user's perspective, how do we choose a method to use, right? That's a very challenging question. And also, if we don't have benchmarking, as users, they will probably go with citations, and then they will probably use older tools rather than newer tools, right? Then as tool developers, that's not good for us, because if we are not the first, then our tools may not be used. And also another question from a statistical perspective is inference. Let's say if we want to infer how a gene is distributed given a cell covariate, it could be the cell's type or the cell's, I will talk about this, pseudo time in the trajectory, the relative location in the latent trajectory, or a cell's spatial location. This is an inference question. Or we may be interested in how every two genes are correlated. These questions require some statistical inference and require some modeling. And finally, for in silico controlled experiments, this is some concept I really want to advocate for computation analysis. And the idea was motivated by wet lab experiment, where we must set up a negative control or sometimes a positive control to make sure our experiments didn't do anything wrong, to make sure we trust our findings. And given that our computational analysis has become more complex, we have multiple steps involved, I feel like this is also something we need to set up from the beginning. So we have negative control or positive control as well to run the pipeline along with our real data. And then we compare the findings to make sure the discoveries we made were not arbitrary or not something invented by the pipeline. So these are the three purposes for our simulator development. So we need a realistic simulator with interpretable parameters to achieve them all. So I want to open my talk by giving this example as a teaser, even though it's not about the single-cell RNA-seq data analysis, it's about bulk RNA-seq data at the tissue level from many human individuals. But I want to show that benchmarking is indeed important, especially when we want to decide whether we want to use popular methods or not. So this was a paper we published in March earlier this year in collaboration with Dr. Wei Li's lab, who is a computational biologist. And his lab used a lot of computational methods for biological questions. So let me just briefly say how we discovered this problem. So in the field of bulk RNA-seq data analysis, a very important task is called the identification of differentially expressed genes, short as DEGs. So the question is, given we have RNA-seq samples from two conditions, we want to compare every gene between the two conditions to see whether the gene has differential expression between the two conditions. So statistically, this is a two-sample testing problem, one test per gene. And EDGE-R and DEC2 are the dominant tools in the field, and their common assumption is that every gene under each condition follows a negative binomial distribution. And because in the standard setting, especially when they were developed in 2014, we usually only have 3 experimental replicates under each condition. So this two-sample testing problem has a small sample size issue. You only have 3 samples per condition, so it's a 3 versus 3 comparison problem. And we don't have good power. And you can see that the t-test wouldn't apply because the data are counts and we don't have the large sample to use central limit theorem. So they assume the negative binomial because the data are counts, and they try to improve the parameter estimation to reduce the variance of the parameter estimates using the empirical Bayes approach. So they could borrow information across genes. That's their basic idea. And what we discovered by accident in that collaboration is that here the data are not experimental replicates, but they are from individuals, 51 individuals who didn't receive the immunotherapy, anti-PD-1 therapy, and 58 people who are on treatment. And the question answered by the Cell paper is to compare these two groups of patients. So if we just run DEC2 and EDGE-R on the comparison, we could find these numbers, like more than 100 and more than 300 differential expressed genes between the two conditions, the red dots. But we found that if we try to set up some negative control by permuting the labels between those patients, among those patients, and we rerun these two methods on the permuted data, wow, we see this distribution as the number of DE genes. We could find from permitted data. So here the bar shows the average and the arrow bar shows the standard deviation. So we can see that from many permitted data sets, we could find more D genes than the method does from the real data. And this clearly shows something counterintuitive. And why would that happen? We look into the data and check the goodness of fit of the negative binomial model. And we found that for the genes that are rarely identified from permitted data, the negative binomial fits reasonably well. But for the genes that are frequently identified from permitted data, the negative binomial fits poorly for each condition in the real data. So in other words, this is a common phenomenon we know in statistics about using a parametric model. We need to check whether the model is reasonable, and clearly the model doesn't fit well for those individual data who are not replicates. So realizing this issue, we want to convince biologists that having this inflated false discovery may mislead their scientific discovery. So here we just took the differential expressed genes found by DESeq2 or HR. On each gene set, we just find which functional terms appear frequently in those genes. This is called gene ontology enrichment analysis. And we can see that many terms are related to immune response, which means that if we trust the results, we may claim between the pre-treatment and on-treatment patients, their immune responses differ, and this is something quite exciting. However, after we realize that these genes will be anyway discovered from permuted data, then the enthusiasm is no longer there because we will doubt the results. So what we did as a comparison is to just take the classical Wilcoxon rank sum test, which is also known as Mann-Whitney test, to compare the two groups of patients. Since our sample sizes here are reasonably large, 51 and 58, I just wonder, why don't we have to rely on the negative binomial assumption? We could just, compare the two samples non-parametrically. And then we see that Wilcoxon is self-consistent. It doesn't find D genes from real data, nor does it find D genes from permitted data. So we don't have that contradiction there. And I have to say that this result has raised a lot of discussion and controversies on Twitter. So if you're interested, you can check what we discussed there. But the very interesting part is that many people became shocked by the fact that popular methods do not always work, and they are very curious about method choices. So that's, yeah.
I'm just wondering, so you have like these two subsets of studies that you have untreated patients, untreated patients, and they themselves will have their own variation in the
Yeah.
Specifically for each gene pocket, there's much more variation.
Yeah.
But then when you're for mutate, are you accounting for this variation? So how is that really for?
We just randomly permute. We didn't consider anything.
Okay.
Yeah, but even so, basically, we just pretend that we don't know which group the patients come from.
Okay.
We just put them together and randomly assign 51 to group 1, 58 to group 2, and run the DE analysis. But regardless of complexity, right? Let's just put it simple. Why would there be D genes after random permutation? Because we didn't do permutation just once. We did many, many times. And here the result is over many, many permutations. So I would say, no matter if you understand statistics or not, this is counterintuitive.
So I'm trying to understand, like, if the variation within the group is too high,
Yeah.
then you will obviously see no, like then the permutation will never work, right? Because in that this case, that within a group itself, there's so much high variation that if you mislabel anything, it will obviously have no impact, right?
What do you mean by have no impact? Because
So if you have literally 100-fold variation within a group, then you're picking up, or if you're picking one sample from the lower end or picking another sample from the higher end, then that means that it's basically the difference between the treated versus untreated is also going to be very small.
that's the result we will get, right? If the within group variance is so big, then you do statistical tests. You need to account for that.
Exactly.
Yeah, exactly. Yeah, that's exactly the reason. And we have some questions on here. Okay. Oh, I'm sorry. Yeah. Okay, thank you for the comment and thank you for the question. That's exactly the reason. When the variance is so big, you cannot say much. Yeah, thank you. Oh, yeah, I remember to repeat the question from the audience. Yeah, but that's just a teaser. As I said, it's nothing about the single cell analysis I will focus on today. So we started this journey for building a simulator for single cell data with this assay design back in 2019. So the goal over there is to have a way for us to estimate the optimal sample size. So here the challenge is that for single cell data, it's not necessarily that the more cells you have, the better. The reason is because you sequence those cells together and the total signal is a constraint. So therefore, the more cells you sequence, the shallower information you can obtain from each cell. So you can see fewer genes will be measured in each cell just because of low signal for that cell. So this is something that's very different from traditional statistics. So we want to see whether we can find that change point in the sample size so that we can optimize the power. So that was the whole idea for this work. And of course, here we only consider that differentially expressed gene analysis, D gene analysis. So we look at one gene at a time. So in other words, we didn't really concern about the gene-gene correlation in this work. However, at that time, we tried to make the model with interpretable parameters, and we should allow the cell number, which is the sample size, to vary, and also the total sequencing depth to vary. So that can help us run some in silico experiments and run the analysis to evaluate how the method works. So these are the pros. And also because of the pros, my other student used this method to benchmark a task. It's called doublet detection method for single cell data, which means that when we run the experiments, it's possible that a droplet, which is supposed to contain only one cell, may accidentally contain more than one cell, and that's called a doublet. And people want to remove those doublets before doing analysis because these are not single cells. So for this, I think when we did this benchmark, 8 methods were developed. So my student used the simulator to generate cells with some known truth, and then we can do benchmarking. But as I just said, there are cons for this simulator. It doesn't capture gene-gene correlations, and also it does not directly model count data. What we did is that we took the gene, let's say gene J's count in cell I. We did some transformation, primarily log transformation, to make the data more Gaussian-like. Then we could use Gaussian model. But I want to say that I don't think that's the optimal way to do it, because how to do the transformation itself is an additional arbitrary step. So motivated by this, we continue to improve our simulator. And when we did our second version, you can see these are the simulators available at the time. And we try to summarize what they can do and what they cannot do. And first of all, I think whether it can work for different single cell RNA sequencing protocols, is it adaptive? That's the first criterion. Does it preserve actual genes? Not always. Some of them may simulate data, but the genes are what I should say invented genes. And do they capture gene-gene correlations? Mostly no. Do they allow the cell number to change or sequencing depth to change? No. And I want to point out this is something interesting, because in our formulation, every cell is taken as a random observation from a population. So that's why the cell number is the sample size in the statistical sense. But in some methods, they treat cells as fixed, just as genes. So then in this case, the cell number cannot change because cells are considered fixed quantities. And also whether it's interpretable, basically we just say, oh, if a method uses deep learning, it's less interpretable. And finally, is it computationally efficient, or does it work when your real cell number is large, right? So these are the properties we consider. In particular, I want to say gene correlation is an aspect missed by most simulators. So motivated by this, we had our version 2 to capture gene-gene correlations. And I will detail this method in the next slide. But the idea is that for real data, we require the cells to be pre-divided into cell types or clusters. And for each cell type, we want to fit a multivariate distribution for multiple genes jointly. Then from this distribution, we could change its parameters based on some user-specified sequencing depth and the number of cells the user want to sample, then we do sampling. We draw sample of synthetic cells from these joint distribution from each cell type. These are our synthetic cells, and then people can use this data for guiding experimental design. Again, what is the sequencing depth, how many cells to sequence, and also to benchmark computational method. And I want to say that while we work on this, we have found similar work that try to capture gene-gene correlations. But I want to say that we do have some advantages. For example, this one doesn't allow a cell number to change, and this work requires some additional input of ground truth, which we don't. Okay, so I will introduce the notations. This is probably the most technical part of my whole talk, but I'll try to make it just not so difficult. So the notations here is that, as I said, here the count matrix is denoted as gene by cell, which is actually the transpose of my first slide, because this is what we have in the paper. And some people do use this orientation. So the P genes are P features and N cells are the N observations. So we assume this matrix contains K cell types, and the cells assignments to cell types are known in advance. Okay, so then let's assume that we have in the cell type small k, we have NK cells. So this is the submatrix corresponding to the cell type. So from here, we will fit one multivariate distribution per cell type. So I would just focus on one cell type only and drop the cell type index. So our idea is to first model the marginal distribution of every gene, and then we want to couple these multiple genes marginal distributions into a joint distribution using the technique called copula. So our goal is to model counts directly, so we don't need to transform our data. Here, this is a vector denoting the cell J. So because we have P genes, the cell J's count is represented by p-dimensional count vector. And the goal here is that within each cell type, which is what we have here, the cells are IID. So they are identically independent observations from the same joint distribution. So in particular, the small xij is the observed count of gene I in cell J. So this is a count variable, and also it's for gene I. So what we aim to do is for gene I, we want to find a proper count distribution given the cell type. And we consider 4 commonly used count distributions: Poisson, negative binomial, and their zero inflated versions. So prior to our work, people have found that we could use one of them to describe single cell data distribution pretty well. So that's why we only choose among the 4. And we use the model fitting to choose the best distribution that fits to real data. So that's the marginal distribution, which is easy. And the key is to find a way to link genes in the joint distribution. And we use the copula framework, which is very nice technique. So the idea is that suppose we already have random variables uniformly distributed in the interval 0 to 1. Can we couple them into a joint distribution? So in other words, you see, if we consider the P genes joint distribution, and this is the cumulative distribution function, short as CDF, we would denote this as big F. So this big F can be represented by this copula function where it has a p-dimensional function. So it maps this p-dimension space, p-dimension cube, 0 to 1 to this 0 to 1 interval. The copula function can allow us to just input every feature after its own marginal CDF transformation. So it is a known fact that if this random variable follows F1, after the transformation, it becomes uniform 0 to 1. So we have P of those, and we link them into a joint distribution. So one technical thing to note is that the theory for copula is only for continuous random variables. So those P random variables must be continuous. But That's not the case in our example because we have the model as counts. So something we need to do is to make them continuous, and then we could use the simplest copula, which is called Gaussian copula. I'll just talk about this first. So what it does is that given the transformation, okay, so this is a uniform 0 to 1. This is a uniform 0 to 1. If we denote them as Uij, U1j to Upj, all of them are uniform 0 to 1. We first apply the inverse CDF of standard Gaussian, univariate standard Gaussian. So this becomes a standard Gaussian random variable, and so does this one. And then we couple them together into a p-dimensional joint Gaussian distribution. And then what we only need to estimate is this correlation matrix, because we assume that every random variable here has mean 0, variance 1. So the only thing to estimate is the correlation in the Gaussian setting. So that's the Gaussian copula. In other words, we separate the marginal distribution estimation in the previous step from the estimation of correlations in this step. And as I said, here our Fi for Jingyi is not a continuous CDF. So the Gaussian copula doesn't seem reasonable. To make the transformation more continuous, what we did is to use the so-called distributional transform, which we did is that taken Xij account, we just take its value of evaluated at the CDF, and we take its value -1, evaluate at the CDF. And between these two evaluations, we use random in linear interpolation. So we just randomly draw this Vij from uniform 0 to 1, is a value between 0 to 1. And then we just multiply Vij, 1 minus Vij as 2 weights. Then this Uij will become continuous. So just that's the way we try to avoid this continuity issue. So together, you can see that with this Gaussian copula, we have a way to approximate the p-dimensional joint distribution of p genes. And with this, we could do sampling. So yeah.
This is really nice because then you are assuming everything is IID, and there's no extra sigma that you worry about, right? There's no extra standard deviation covariance that's happening between different genes. So,
No, we are not assuming genes to be IID. That's the key. The genes are called cells are IID. Yes.
But each U1 is a cell, right? Or it's a feature.
U1.
So the phi inverse of U1.
So okay, so Xij is the count of gene I in cell J. And basically, this transformation, if you ignore the distributional transform, Uij is just equal to Fi Xij. We just apply the marginal CDF transformation to make Xij into Uij, and Uij is uniform 0 to 1. That's the only thing we did. Yeah. So the reason is because the copula function works for uniform 0 to 1 random variables. That's because of it. So basically, you don't have to estimate Fi and R together. We do this in a two-step estimation. This is exactly what we do. So here is the estimation. So we denote for gene I, Fi hat as the estimated marginal distribution, and this could be Poisson, this could be negative binomial, or it could be their zero inflated versions. And then with this, you see, we will apply this Fi hat to transform our observed count, Xij, count of gene I in cell J. And to make it continuous, we apply this distributional transform technique, Vij sampled uniformly from 0 to 1, and then we do this to obtain Uij. And finally, to obtain an estimate for the multivariate correlation matrix, we have to transform U1j to UPJ by the inverse of standard normal, right? So then marginally, every gene I is standard normal. And then to estimate their correlation, we just have every vector for every cell, and that will give us the input. Yeah. Exactly. Yeah. higher order. Exactly. Empirically, exactly. So empirically supported, because actually we found that by saying empirically supported, we found that our synthetic cells sample from this distribution mimic real cells. Yeah, thank you for the question. Yeah, I think that's a very nice point about the higher order interaction. Yes. Okay, so to make it simple, what do we do with data simulation? After we fit the marginal distribution for every gene and the correlation matrix for multivariate normal, what we could do, and we do that for every cell type, right? So then what we allow users to input is the number of cells they want to simulate and the total sequencing depth they want to simulate. So this parameter is used to change the marginal distribution of every gene, to change the mean for Poisson or negative binomial. Finally, we will output a synthetic cell by gene by cell count matrix for each cell type with a total of k cell types. And we will also output the model parameters we fit. So this is the result. So based on what I said, you could see that this is the data we used to fit the model. And these are the cells we reserved, we didn't use for model fitting. And this is the synthetic cells we generated. And we check how well it mimics the real data using this measure. And it's short as, I think it's complex, like something like mean, local, inverse Simpson index. So this measure we can simply interpret as for every cell, we construct a neighborhood in the principal component space. And within the neighborhood, we count how many neighbors are from the real cells, how many are from synthetic cells. If they are evenly mixed, this number should be close to 2. Otherwise, if they're random, not alike at all, the number should be close to 1. So the range is 1 to 2, with the larger, the better. So this number will show on average how well do the synthetic cells mix with real cells. And you could see from this figure that the copula plays a significant role, because if you don't use it, just a marginal distribution, the cells won't look like real cells at all. And these two are two existing simulators which do not explicitly model gene correlations, but they have their own way to mimic real data. And I also want to point out that they consider cells to be fixed. So whenever the training data has n cells, they generate n cells. They cannot generate different number of cells. So we see that our synthetic cells are more similar to real cells. And this is our paper that describes the details of the simulator. But one thing I want to mention for biologists is that the limitation is we are fitting a model per cell type. What if the cells don't follow a cell type? What if the cells cannot be well clustered, right? These are the questions we need to address. So that's why we move on to our version 3. IICD 3. So in this newest version, we could do the following for simulation. So as in our IICD 2, the cells could be in discrete types. So in other words, in this sense, IICD 2 becomes a special case of IICD 3. We also can simulate cells along continuous trajectory, or we could simulate cells from a space, like a 2-dimensional space or 3-dimensional space, spatial data. And beyond RNA-seq we generate from IICD 2, we could generate other feature modalities, like I said, ATAC-seq for open chromatin, site-seq that gives us both protein and also RNA expression, and also DNA methylation. Finally, this is something we newly added for experimental design. So we could generate data sets in batches, and they can have different covariates or from 2 conditions or from 2 batches. So we could specify these in our parameters so that we can generate data according to the design. So this is just to show that how it improves when the data follows a continuous trajectory. So you can see that using IICD 2, we have to force our cells to be pre-clustered and then generate the data. But with IICD 3, I can roughly say that we replace that per cell type model. We generalize that to be a regression framework. That's how we modify the marginal distribution of every gene. And then you can see that we can generate cells in a continuous trajectory. And finally, to make sure that we could output a likelihood for some interpretation, we use the technique called Vine copula that allows us to decompose the copula, the multivariate copula function, into a product of bivariate functions. And this can allow us a way to obtain a likelihood, but it's an approximation. Okay, so that's the continuous trajectory. This is a new functionality for spatial data. So for spatial data, what we did is to assume every gene's mean expression in the space follows a Gaussian process specified function. So this can give us smooth function. And on top of that, given the mean, we could assume the randomness follow negative binomial, and then we could mimic the real data pretty well. And also we could output a mean function for interpretation, which I'll show. And finally, we have this additional functionality thanks to my other student who led a parallel project. So we could go from the synthetic counts we generated to generate synthetic sequence reads. And this will give us a way to benchmark lower level bioinformatics tools that don't start from counts, but from reads. So that's the simulation functionality. And for the interpretation functionalities, we could output some model parameters for interpretation. Like if we assume the cells follow clusters or cell types, we could give you the mean of every gene in the cell type. And we could give you the mean function of a gene that changes along the trajectory, or how the gene mean changes in the space. And as I said, using the Vine copula technique, we could give you an output of the likelihood. And based on the likelihood, we could calculate the Bayesian information criterion. So this advantage is that given our model will use some user-specified pseudotime as input, we could give you the quality of the pseudotime or clusters without having ground truth. So the idea is just to assume, given the clusters as this, we use it to fit our model, and then we evaluate how well does the model fit to data. So that's reflected in the BIC, and the model complexity is also considered here. So finally, I want to emphasize this model alteration functionality. So given how we fit the model to real data, we could change the model parameter to specify our hypothesis. So in this example, we could change the gene's mean parameter in the two cell types to be equal. Then the gene is known to be not differentially expressed. And this way, using this parameter setting, we can generate synthetic cells for benchmarking. And finally, this is an example where the real cells may be in two cell types, but if we want to set up a contrast by generating cells from one cell type, but mimic the real cells, what we could do is that we could fit our model, assuming there's only one cluster to this data, and then fit a model to generate those synthetic cells. So they will only have one cluster. So this idea is very similar to what we called the likelihood ratio test in statistics. So we basically fit the model under the null hypothesis, but here we are not giving just a likelihood, but we can give you synthetic cells by sampling. So these are the things I just mentioned. We could tell you this is the function we fit for every gene along the pseudotime in the trajectory. So the technique here we use is basically spline to get the nonlinear function. And this is by Gaussian process. We can get the gene's mean function in two-dimensional space. And these are the correlation matrices from our Gaussian copula. So you can see that we actually can give you different correlation matrices estimated for different cell types. And it actually reflects the cell type similarity. And using Vine copula, we could approximate this correlation matrix using a Vine structure. And the interestingness for this is that we could interpret different layers of the Vine for different conditional correlation. And the first layer is just the strongest marginal correlations. So I think biologically, this may have some interesting interpretation.
And as I said, we could use BIC to assess the quality. So here we have 3 trajectory inference algorithms used in the field. And we found that if we inspect the BIC output by our model using their corresponding pseudotime, we can see Slingshot has the best BIC. And visually, we can also see the pseudotime it gives for those cells agrees with our eyeballing the best. So I think at least it makes sense to say, okay, our results are reasonable. And finally, for model alteration, as I said, if this is how a gene's expression has in real data, so here we have 2 cell types. Within each cell type, we perform some stimulation. So this is control stimulation. So you can see that this gene has elevation expression level in both cell types. And if we don't change anything, we just use the fitted model to generate synthetic data, synthetic cells, and this is how it looks like real data. But we could manually set this gene to have the same mean parameter before and after stimulation. So then we could see that here the gene doesn't change in both cell type. Or we could just change one cell type and keep the other as in real data. Then we have this other data. So we could modify the model parameters to do many things. And this is another thing because in our model fitting, there's a parameter for batch effect. So if we fit our model to real data, given the 2 batches, and the synthetic cells will still have 2 batches, you can see the cells are not mixed in the 2-dimensional UMAP space. But once we remove the batch effect parameter in our fitted model, then we generate synthetic cells, then the cells are mixed. And finally, this is what I said, given the real data, we could fit a model with 1 cluster, and then the data will look like real data because gene-gene correlations are still there, but we actually have no clear gap between the 2 clusters. So everything I said is in this preprint paper, which we have posted, and we welcome comments and also users' feedbacks. So finally, I want to say why we think having this in silico control experiment is so important. This is from the Khan Academy cartoon showing that in wet lab biology, we do need a real negative control group to really conclude whether our treatment makes a difference. And this is also the same as in the clinical trial, where you need control. But in single cell, I think one challenge that has been mentioned by many researchers is this double dipping challenge, which means that we use data for more than once, and then our previous step will affect the final statistical inference. So I will quickly go over this problem called pseudotime DE. So the idea is that the cells we observe in single cell data don't have a latent time variable. But we want to infer the latent time variable to show some dynamic process. For example, it could be cell differentiation, or it could be immune response. So this is a cartoon from Slingshot, a common popular pseudotime inference algorithm. So this is PC1, PC2, first 2 principal components, and the colors mean cell clusters. So given the cluster centers, it does a minimum spanning tree algorithm to link them into a tree, and it smooths out the tree branch using principal curves. And finally, every curve is considered as one trajectory, and the cells are projected to the nearby curve, and the projections are considered pseudotime. So the idea is to order cells based on their gene expression similarity in the hope that the trajectory will reflect underlying biological process, even though we only observe a snapshot of these cells. Okay, so given the pseudotime, a natural downstream analysis is to ask which genes have expression changes along pseudotime. So this is a gene, we can see it's an immune response related, and the pseudotime here is from real data, and we know it tried to capture immune response. So clearly the left gene is elevated and should be called a DE gene. The right gene is not. So we want to find this statistically. And existing methods try to use regression to capture the change, but the fact here is that pseudotime is not a real time variable we observe, but something we inferred. So this shows the uncertainty in pseudotime. We just did this using subsampling to show that yes, sampling uncertainty will indeed reflect be reflected in the pseudotime value uncertainty, and this should be considered. So to address this issue, we propose this method called pseudotime DE to consider this double dipping. And our idea is very straightforward. So given that existing methods use the inferred pseudotime and use regression, so to make it nonlinear, we consider the generalized additive model, which is more general than generalized linear model because it allows the covariate effect to be nonlinear after the link function transformation. So we use this method as in an existing method, follow the approach. But our difference is that to compute the p-value, based on this regression test statistic, we didn't use the theoretical null distribution because the reason is here the pseudotime is not real time. It's not reasonable to consider it to be fixed. So our strategy is to use this upper loop to consider pseudotime inference as part of our procedure. So in other words, the randomness is carried on, and also we use permutation in the middle to disrupt any relationship between this gene and pseudotime so that we can be sure the test statistic values from these samples are null values. So this is the simple idea to numerically get an empirical null distribution for the test statistic. So we are not really relying on the theoretical assumption. And we could see that this indeed gave us more well-calibrated p-values. And Tracy is an existing method. It used the GAN model, but it didn't calibrate the p-values. So they had some deviation from the uniform 0 to 1 distribution for null p-values. And the consequence we have here is that surprisingly, we could find more D genes that are functionally important. So some Go terms, functional terms, are only enriched in our D genes, but not in the existing method, which is a very happy surprise. So I want to quickly say that even though we had this method, but we know it's not perfect. And it cannot address the issue that what if the cells don't follow a trajectory at all? This we call complete null hypothesis. If that's the case, then that testing procedure we designed would fail. Even though I won't talk about the detail there, but this is a fact we know. So the question is, how do we generate another set of null sample that reflects the complete null? And the simulator I described can offer a solution. And second is about the computational time issue, because our numerical empirical null idea will require many rounds of subsampling pseudotime inference permutation. So the p-value resolution depends on this number. Let's say we want a p-value of 0.01, we need at least 1,000 rounds, and that can be time consuming. So we want a way to be able to still control the FDR without using high resolution p-values. So these are the questions we have remaining. And the related question you can think of is the cluster DE problem. You use the gene expression data to define cell clusters, and between the clusters, you identify DE genes. This is also double dipping because you use the gene for twice. So there are 3 existing papers that try to tackle this question. They require either Gaussian distribution for gene distribution, or they require Poisson distribution for gene counts, and also use this interesting idea, count splitting, to make sure that you avoid the double dipping. So I won't get into detail, but this is our analysis of existing methods for avoiding double dipping, either splitting cells, splitting genes, or splitting counts in this paper. But interestingly, when genes are independent, yes, gene splitting, count splitting can give you uniform p-values under the null. But when genes have correlation, the problem is still there. So motivated by this, we want to use our simulator coupled with an idea we borrow from the knockoff paper in statistics to achieve FDR control with synthetic null data generated by SN3. So I want to acknowledge the gap statistic paper for the motivation as well, because it gives us the idea that, oh, to find the number of clusters in this paper, we need a null control, and which is the same idea we use here for setting up a null control. So just to give you a quick idea here, permutation of labels wouldn't work because if the real data does have clusters, and if you permute the labels, the cluster structure is still there. But what we need is a control where the cells don't have a cluster structure. Okay, so I will just quickly say that the Clipper relies on the FDR control theory from the knockoff paper. So essentially, we no longer rely on p-values. What we only need is just a way to construct a statistic for every feature. And here every feature is a gene. So the statistics, we refer to them as contrast scores. And the theoretical condition is that the contrast scores from the null features, which are not interesting, should be symmetric about 0. In other words, the contrast score absolute value should be independent with the sign. So with this assumption, then this idea will give us a control for the FDR. Basically, the right tail is our discoveries. By symmetry, the left tail is the false discoveries, and we want to control this ratio under, say, 5% FDR threshold. So the idea becomes we don't need to worry about the null distribution or p-values, but just designing contrast scores. So in short, this opens up a lot of doors for constructing contrast scores because the target data is what we're interested in, and the null data is what we want to generate or we can do by permutation. So as long as it's properly set up, we could do the contrast. And here the T can be a summary statistic or even a complex pipeline. We just apply it to real data of our interest and null data by simultaneously, and we do the contrast. And then hopefully, if your null data is set up properly, if a feature is not interesting, this difference will be equally likely positive or negative. So it satisfies the assumption of symmetry about 0. So this is the Clipper idea. I won't go into the detail, but I want to say that apply this to the cluster DE problem. We could see when the real data has one cluster to our visual inspection. If we force ourselves to do clustering, like using popular SURAT or k-means clustering, we could always force ourselves to get 2 clusters. And of course, here 2 clusters are driven by certain genes. And then of course, given the clusters, when you find which genes are different, we will find some genes, right? And these genes are not of our interest because there's no cluster at all. But if using our simulation idea, here the null data, assuming 1 cluster, it will look like real data. Then you could imagine whatever pipeline, SURAT, or k-means you apply to both real data and null data, their findings should be similar. So that contrast will work. And so this is indeed the case. Using our idea, we could control the FDR reasonably well, but the naive approach that ignores double dipping, you apply clustering, and then t-test, Wilcoxon test, etc. You will have an FDR exactly 1 because every discovery you made will be false discovery. So take-home messages, I'll stop here, and my take-home messages will be, first of all, I think computation analysis requires sanity check. And popular methods do not always work. This is something we have to bear in mind. And also benchmarking against classical methods is very important for our method developers. We need to have solid motivation to develop new method and see what gaps it can fill. And also our simulator, which I spent time to talk about, can be used for benchmarking, parameter inference, and in silico control data generation. But of course, we have a lot of things to improve, and also I'm very open to collaboration or user feedback about how we can do better with the simulator. And double dipping is a very important problem for statistical inference because in especially single cell and spatial omics data, we don't use data directly. We have a lot of previous steps to give us variables for the final step. So given that, we have to consider those issues. Otherwise, our statistical inference may not be meaningful. And finally, we propose the simulator to help us generate data from the specified null, and also Clipper for making sure that FDR control is easier because we no longer need high resolution p-values. So related to this, I also wrote a perspective article with my collaborator at USC. So we talk about this inference versus learning problem, especially this article is written for practitioners, because we know that even to us, maybe testing and machine learning classification seem very different, but they may actually confuse the two because both are making some binary decisions. So if you're interested, you could check it out. So finally, I want to thank my group members, former students, Vivian, for leading this effort, first effort, and then my fifth year student Tianyi for making the second version, and fourth year student Dongyuan for continuing the efforts as design 3 and the pseudotime DE I briefly talked about, and Guangao for making the read generation possible, and finally Xinzhu and Kexin for doing the FDR control thing and to make it possible that we don't need to worry about so many rounds of data generation or computational efficiency. And of course, the funding agencies for their support. So I look forward to your questions and thank you for your attention.
Thank you for the talk. It was very interesting. Do we have any specific questions? Go ahead.
Sure. So you mentioned some additional issues with the
Yeah, permutation.
Okay, but for the knowledge, which are most of the people, would you be able to reject an extremely value just by say 100 permutation?
We could, we indeed could. Yeah,
Do you want to repeat the question?
that's a very good question. So the question is for the null genes, we should be able to reject them without many permutations, right? I think that's your question. Yeah, I think that's exactly right. So that's why when people use p-values for FDR control, like the standard way is to use the Benjamini-Hochberg procedure. Yes, FDR is control, but what you lose is the power. If your resolution is not good, then for some genes you are not totally sure, you may tend to reject them. Right, so you would reject more and lose power. So that's why resolution is very important for good power. But what we aim to say here is that this theory allow us, actually has been analyzed by some statisticians already. So using this idea, not p-value, but just directly thresholding the contrast scores using this idea, it can give us better power. And you can only do one permutation or one null generation, and that can be good. Even though we're still working on this idea about whether we could find an optimal number of null data generation to optimize the power. And it's very interesting. It's not necessarily the more the better, because there's a balance involved in this comparison. But yeah, we're still investigating this technical issue. Thank you for the question. And we have a question from the chat. Okay, it's from Lawrence. Yeah, please.
So maybe it's a naive question, but how do your methods work in a very complex data when you have multiple samples, you have to integrate across tissues, across patients, and then there is a continuous distribution, and then you have discrete populations at the same time?
Yeah. That's a very good question. This is the question we indeed try to tackle in this version 3. So I didn't show the detailed model because of time constraint today, but if you are interested, you can check our paper. So right now we consider, so basically let me quickly go back. Sorry. I quickly go back to this part. Maybe I can explain with this. Right, so here for this Fi, right, marginal distribution, this Fi. To estimate this, now in our new version, we allow the mean parameter of gene I to depend on the cell covariate. And the cell covariate, as you said, it could involve batch, condition, those discrete covariates. So these will enter the mean function as intercept effects. And then we have something additional to model on, like the space or like the pseudotime. So these are the non-linear part of it. But those covariates, like our discrete, will enter as intercept effects. So then, but at least in our experiments, we found that the model can fit it reasonably well. And then when we want to remove the batch effects, we just set the batch intercept effect to 0. Yeah, thank you. Yeah. That's a very good, yeah, exactly. We use our assay design 3 to generate null data. We didn't use the knockoff to generate null data. Because in the knockoff paper, the key part is how to generate the knockoff, right? That's their key, but we didn't follow that procedure for several reasons. First, our question is different. So in their question, it's a multivariate prediction problem. They want to make sure in the knockoff, right, every feature is conditionally independent with the response given of the features. That's not the question we're examining here, because all the questions like the DE genes we try to answer, we examine one feature at a time. So that's the key difference. So we don't feel we need that go that far. Yeah, thank you. And that makes the problem actually much easier. Yeah. Yeah. Model alteration, yes. Oh, why do we want to modify gene expression? Right. Oh, that's a very good question. Because if the null hypothesis, let me just go back. Okay, go to here. Go to this example, for example. If the null hypothesis is not that there's no clusters, but just that the clusters are there, but one gene is not differentially expressed, then we could use that way to generate the null data. So we still fit the data, fit our model to data by assuming there are two clusters. But after we fit the model, we set one gene's mean parameter to be equal. Yeah. Yeah. Thank you. Yes. Mm-hmm. Yeah. Uh-huh. Oh, I see. That's a question. The question here is, how do we do this? The goodness of fit. We just do, actually, there's an R package for checking this, for checking the goodness of fit of a negative binomial. But yeah, you made a very valid point. We know that here the p-values are not completely valid. So if you found you want to be really rigorous, there's no issue for negative binomial for checking the goodness of fit because it has two parameters, the mean and dispersion. But anyway, we still try to show that qualitatively the degree of fitness change or differ between the genes that are rarely identified from permuted data and the genes that are frequently identified from permuted data. So the goodness of fit does show some difference. Yeah. Oh, great. Okay.
I think we're done. Thank you. Thank you for all the comments and attendance. And we'll see you next month on the next meeting. Thank you.
Thank you, Carol, for the introduction hosting. And thank you everyone for being here.